It's Jupiter. I knew that. I was testing you guys.
What the fuck?
Hi, everyone. Can you hear me loud and clear? Yeah. Okay, I got some responses in the chat. It's fantastic. Okay, so I guess let's just get moving. I think a lot of the class is still not on, but this is we have quite a bit of materials to go through, so we'll just get moving now. Okay, so it says today we're going to be using iClicker and CrowdMark. We're not using CrowdMark today. We're just going to use iClicker for this activity session. Just a reminder that the final take-home deep questions are going to be released tonight at 7 p.m. You have until Tuesday, April 11th to complete those final deep questions. And Thursday's class is going to be a work session where you can work on them together if you wish, or by yourself. It'll be a work session. It's due Tuesday, April 11th. I think there are only 4 of these final deep questions, and they're very similar to the deep questions that you've had so far. So anyways, they're due April 11th, so Tuesday the following week. So you have 1 week to complete them. Everyone gets 1 grace day, meaning that you have up until Wednesday, April 12th to hand them in. And that's regardless of whether you've used your grace days for the other deep questions or not. you have that one, everyone has that one grace day. Okay, so don't leave it until the last minute. Requesting, I tried to submit in the last minute and it wasn't working, all that kind of stuff. Like you have to submit it before the clock ticks 11:59 p.m. Okay, so don't leave it till the last minute. I forget what else I wanted to say about that. I think that yeah, I think let's just move on. But again, they're very similar to what you've seen before. There's also a survey that you should do. It's due Wednesday. So this Wednesday, it's in place of the activities that you guys normally do that are due on Wednesday. So please be sure to complete that. I'm going to post quiz grades soon on eClass, just so you can look them over. Again, we dropped the lowest 2 quiz grades. Soon after that, I'm going to post the assignment grades. If you see any issues with the grades that are posted on eClass, send a polite email to bio2040 and I will do my best to check into it and get back to you. Okay? All right. So let's move on to today's lesson. Okay. Yeah, so today we're going to talk about African elephants. Sorry, I'm having some technical problems here. There we go. Today we're going to talk about African elephants, and specifically elephants in the Gorongosa National Park in Mozambique, Africa. These beautiful animals, as you can see here,
I finished the chapter.
Majestic animals,
and I would like to
have tusks. At least some of them have tusks, and some of them don't.
That way, we can finish the game basically.
And the tusks are actually used for defense. They're used for digging, lifting, stripping bark off trees. That's just something that elephants do, but actually it's a very important thing and creates environments for organisms, certain niche of organisms to survive. Okay, so the tusks, they protect the trunk, they protect the elephant, and they're a valuable tool. Like digging is important for elephants. In order to find water, sometimes they need to dig in the ground, so they use their tusks in order to do that. One thing, though, is that the tusks have become a valuable commodity worldwide. Not become. They've been a valuable commodity for centuries, and therefore people have hunted elephants that have tusks. They harvest the tusks and they make various things out of it, as you can see here, like jewelry and so on. Okay, so elephants with tusks have been heavily hunted for the past few centuries. And so over the past 100 years or so, what would you expect to see? So I guess take a moment to think to yourself, given the information that the ones with long tusks have been heavily hunted over the past few 100 years, what would you expect to see happen to the population of elephants? And so, I guess you could probably think of a few things, but you would expect to see which of the following. Hold on a second here. I just need to bring up the clicker.
Professor, I also don't think you're recording.
Oh, geez. Thank you.
Recording in progress.
Okay, so thank you for the reminder. I forgot to hit the record button. And also forgot to log into Clicker. A little under the weather today, so my brain's not 100%. Okay, so just to recap for those of you that are watching the recording, we're looking at elephants for this activity and particularly the tusks. We're going to look at the genetics of of tusks, and the tusks are very useful to the animals, but also they have been heavily hunted, and all the elephants that have tusks have been heavily hunted for the past few centuries. And so the question was, well, what do you think is going to happen to the population of elephants since the ones with the large tusks have been heavily hunted for the past few hundred years? Okay, so I'll start the clicker now. Go ahead and submit your guesses. And don't be shy. Again, it's just for participation, right? Okay, so just a few definitions to help you. This might help you out, but in case you don't remember from Bio 1000, stabilizing selection is when a population is pushed toward like an average or a median trait, okay? And directional selection, on the other hand, is when a population is pushed toward one extreme phenotype rather than the mean or the opposite extreme. So 5, 4, 3, 2, 1. Okay, so most of the class, 46%, is saying E, a decrease in tusk length in elephants over the past 100. You would expect to find a decrease in the tusk length of the elephants due to directional selection. Okay, so there is a strong selective advantage to having smaller tusks or no tusks. So in African elephants with large tusks, the ones that have the large tusks, they've been heavily hunted for the past few centuries. So what you would expect to find is E was the correct answer. You find a selected advantage of having smaller tusks. And so you'd have a decrease in tusk length in elephants due to directional selection. Okay, so just to clarify, a few things. It's not going to cause the appearance of a tuskless elephants. They already exist. So what's found in the population is a large variation in tusk length. Evolution is not goal-directed, so hunting the elephants with large tusks is not going to cause the appearance of a new trait like the tuskless elephant in the population. However, it would favor smaller elephants with smaller tusks and elephants without tusks. So, so the populations vary in terms of their tusk phenotype, right? And if the variation has a heritable component, then it can evolve. And so if the elephants that have the large tusks, like the one we're seeing on the left here, have been heavily hunted over the past few centuries, there's a heritable, this would cause a difference in inheritance, right? So imagine you're eliminating all of these elephants that have the larger tusks. Then there's no the genetic variation that gives rise to that large tusk size would slowly not exist in the population anymore. So you'd still get your variation like you normally see. However, these beautiful ones here would be eliminated from the population. So on average, you'd see a decrease in tusk length in the elephants due to a directional selection. Okay, so we're going to look at the rise of the tuskless elephant. So in Gorongosa, Mozambique, they noticed, this researchers, they noticed a significant increase in the tuskless elephants over the last 30 years. And specifically, tuskless female elephants. So you can see here, no tusks. These are females with no tusks. And so the increase was from 18.5% at the beginning of the 30-year period to the tuskless females ultimately represented 51%. So they went from 18.5% to 51% of the population over that 30-year period. And interestingly enough, it's specifically the female elephants that are being found to be tuskless. There are no evidence of tuskless males, which is kind of odd. Why are we seeing no record of tuskless males? Like, what's happening here? Okay, so would you hypothesize that the genes responsible are sex-linked? You're not seeing tuskless males. You're only seeing tuskless females, right? Five, 4, 3, 2, 1. Okay, so 84% are saying yes. You would hypothesize that it's sex-linked. Again, it's not found in the males, only the females, right? Okay, so one of the things I want to mention here, it's important when if you think you have a sex-linked gene, it's important to actually keep track of your chromosomes whenever you're looking at inheritance patterns. And so, you know, that can be typically done if you're to write like your X chromosomes, something like this, and maybe it could be a plus, like it has the allele that has the trait. Maybe tusks would be represented by having the trait, so X chromosome with plus for that particular allele. And if it didn't have a trait, it could be X chromosome minus. Now, males, on the other hand, elephants are similar to us. They have an XY system. Okay, so we'll look something like this. There are no, as we mentioned, there are no tuskless males. So the only allele that you would find here is X plus. Okay? You want to follow you want to draw your chromosomes, you want to follow the particular phenotype that you're looking for. You got to remember, this is a wild population of elephants, and we can't like really do controlled breeding, that the scientists weren't able to do controlled breeding. So all they could do actually was to follow the population elephants, follow the phenotypes and the proportions of phenotypes. They could follow certain groups, and they could kind of get an idea of which ones were mating and so on and so forth. But for the most part, they could only look at the percentages of elephants that are male and female, and the ones the percentage that had tusks. All right, so we're hypothesizing that at least some of the information important for tuskless phenotype is actually on the X chromosome, because like you told me, it's sex-linked, and it's sex-linked, and particularly the females are showing phenotype.
Jeez.
Okay, so let's hypothesize that at least some of the information for the tussless phenotype is on the X chromosome. So thus, a tussless female could have one of two phenotypes. Actually, it can only have one of these two, genotypes. So again, so we're gonna annotate if the allele is thought that it contributes to tuss formation, it's gonna have the plus sign. So X, and we got our plus sign here. If it's the tussless phenotype, on the other hand, it's gonna be the minus sign for no tuss. Okay, so the tusk information on one X chromosome and tussless information on the other X chromosome. That would be this particular genotype here. And we have the tussless phenotype shown here, where we have the tussless alleles on both chromosomes. So I guess I might be getting a little bit ahead of myself here, but
Wait, what'd you do?
yeah, we'll come back to it. I want to get ahead of so therefore, a tussless female could potentially have two different genotypes. What's the probability of a tussless female and a tussless male having a living female offspring? Living female offspring.
Mm-hmm.
5, 4, 3, 2, 1.
I don't know what happens.
Okay, so most of the class is saying C. Okay, so what's the probability of a tussless female and a tussless male having a living female offspring? This is I guess it's a little bit of a trick question, but remember that you would be right. If everything was normal and, male and female inheritance with the XX and the XY chromosomes, the offspring has 50% chance to be a male, 50% chance to be a female. However, we did say that there were no tussless males. Actually, this cross is not even possible, right? A little bit of a trick here, but it's not possible. So it would be 0% the probability that the tussless female and the tussless male have a living female offspring, because the tussless males, they don't even exist.
Bro, how you getting these, bro?
Okay, so males always have tusks. So you assume that they don't have any tussless information. How to write the genotypes of males? Males are, I'm not going to do a clicker here. I'm just going to tell you the answer. So males, if you had a tuss information on the one allele, you would have the X with the plus sign, so indicating tuss, right? And the Y chromosome doesn't encode anything. Okay, so that would be for the male, right? And now, if you think about it, when we go back to those crosses that we were talking about earlier, where does this one X chromosome come from? And this X chromosome here, where does it come from?
Mm-hmm.
Anyone want to unmute yourselves and let me know? Okay, I guess not.
Yeah. I guess X plus should be from the let's say from the male, and then X minus will be from the female, because the one of the alleles of the male is always X plus.
Excellent. Very good. Yeah. So the male can only contribute the X plus, right? So that means the X minus. The male can only contribute the X plus because the males can't be tussless. They have to contribute the tuss information. And the female, on the other hand, could contribute the X minus, so the tussless genotype allele. So, and if you were to look at this cross here, is this cross even possible? Or sorry, this genotype. Is this genotype possible? Yeah, go ahead, Dinara.
No, do you hear me?
Yep.
No, there is no possibility of having X minus because we don't have tuskless males.
Exactly. So, because one of these alleles has to come from the father, and
Only
ultimately
male
the father can only be
elephants have
X
tusks?
plus. Great. Fantastic. Okay, so the males are
not all of them
always X plus 1. What's the probability of a tuskless female and a tusk male having a living female offspring?
I have a question.
Sure.
I'm not sure if I missed this, but why can't a male inherit an X minus from the mom?
Because there are the tuskless males don't exist.
Mhm.
And since the male can only have one X chromosome, if it had an X minus, which would be tuskless, it would show the phenotype. But they don't exist, and therefore this is the only genotype that can exist. Good question.
Is there a reason why they don't exist? Like why they can't inherit it from their mom, the X minus?
We'll get to that.
Okay.
That's what the activity is about. Yeah.
Okay.
Good. Yeah, no, that's good. You're thinking ahead. Excellent. Okay, so let's 5, 4, 3, 2, 1. And so 60% are saying C, the probability of a tuskless female and a tusk male giving rise to live female offspring is 50%. And that's what you would think, right? That's what you would think would be the case, assuming that all inheritance is normal, right? 50% male, 50% female. However, in Gorongosa National Park, they were not observing this expected ratio. So they would actually find a significant deviation from the ratio. Okay, so I just want to, for those of you that are maybe not clear, how could we figure this out? So how do we come to the point like the 50-50 inheritance? You have a tuskless female, and it's going to mate with the tusked male, and ultimately you would do your Punnett square. Okay, so this here is your female, and then here is your male, and ultimately they would give rise to these various alleles. So we're not talking about the tusk or tuskless. They're just talking about what proportion are male and which proportion are female, so that would ultimately give you 50-50. And like we said before, I showed this cross anyways, but this genotype here for the females cannot exist because one of these alleles has to come from the father, and we don't from the male. And we don't see any males that have the X minus phenotype. They all have the X plus phenotype. So this would be the ratio that you see. But again, it's a 50-50 ratio of male and female. Okay, so what did they observe in Gorongosa National Park? They actually found that tuskless moms produce about 66% living female offspring, and only 34% living male offspring. And again, all of these males are tuskless. Okay, so it's a significant deviation from the 50-50 inheritance pattern that you might expect. And so they're missing these tuskless males. Why? Why are they missing the tuskless males? So why are we seeing no record of tuskless males? After looking at this particular ratio and thinking about these particular Punnett squares, can you come up with a reason why? Okay, so tuskless moms produce about 66% of living female offspring and 34% living male offspring. So essentially, this is it's skewed. And since there are no tuskless males, we can eliminate them from the equation. Okay, so those tuskless males that were drawn there, again, they don't exist. Okay, so it would be eliminated
You're
from the
so smart.
population. So if this genotype was possible, maybe there's some way, we're not giving up on it just yet, even though we said that the male can't possibly contribute one of these X minus tussless phenotypes. We're still going to monitor it just in case there's something we're missing. And so if this were the case, so if these were the possible crosses, so you have your tusked male breeding with this genotype of tussless female, and this tusked male here breeding with this homozygous tussless phenotype for the female, this is essentially what we would get. And if you look at it,
Oh, you're so smart.
you would have one homozygous tusked female to 3 heterozygous tusked and tussless female. Now, that's not what we're seeing. We're seeing 66%. So we just eliminate those males that are being lost, right? Because they don't have the tussless phenotype doesn't exist in the males. We're still not coming to the 66% of the females being living. Actually, what we get, assuming that this, I think we can only assume that in this particular circumstance that X minus the tussless phenotype would be dominant, right? Because we would have this form tusks, and we don't have any other genotypes to form tuskless. And so the tuskless must be represented by X plus and X minus being a heterozygous, so the X minus is dominant. Okay, so the tuskless moms, however, they produced female offspring that were 50% had tusks and 50% were tuskless. Okay, so I think that's another key to our equation. We already did say that this particular allele could not be inherited from the father, so this actually doesn't exist. If we remove that from the equation, now we get the 50% tusked and 50% tuskless. Those are the only two genotypes we can get. So this must be tuskless. Tuskless must be dominant. Okay, so we get a 50-50 ratio for the females that are produced. And if you look at the ratio, the ratio of the 66% and 34%, so 66% are living females and 34% are living males, that also follows this inheritance pattern. Because if you have 2/3 that are females and 1/3 that is males, that's essentially 66% female and 34% male. Okay, so it appears as though this is the type of cross that we're dealing with, and that the tuskless phenotype is actually a dominant sex-inherited trait. Okay, so is the tuskless phenotype acting like an X-linked dominant trait, or is it X-linked recessive trait? I guess I kind of already answered that, but let's go ahead and see how many people agree or disagree. Okay, 5, 4, 3, 2, 1. Okay, so 71% said A. It's actually a dominant phenotype. So how do we know that? There's 50% of the females are tusked and 50% are tuskless, and we only have these two possible genotypes. Okay, so therefore this tuskless allele must be dominant. Okay, so why do we have no tuskless males? We can have tusked females, we can have a tusked female, we can have a tuskless female, we can have a tusked male, but we cannot have a tuskless male. And so that actually suggests that whenever this X minus allele gets paired up with a Y chromosome, it actually is lethal. And that's why we're never seeing any of these tussless males exist. Okay, oh my goodness. Oh. I'm sorry, just give me a second here. Just one moment. I think my slide deck got messed up. Okay, I'm probably gonna have to skip around a bit. I don't know what happened there, but my slides are all out of order here. Okay, so is the tussling phenotype acting like an X-linked dominant trait or an X-linked recessive trait? You guys told me it's a dominant trait. And then so we were going on to figure out why are we not seeing any of these tussless males? They actually don't exist. And so one explanation can be you look at development in your pre-class materials, right? And they discussed all the various stages of embryo development and how, you go from a zygote to a multicellular embryo, and there's a number of program steps, right? And embryo lethality essentially is whenever you have a particular, in this case, a particular genotype that actually it causes one of the steps of the development to fail. Okay, so that could explain why we're not seeing, if we're only monitoring the number of living male and female offspring that are tuss and tussless, if any time we have the formation of that X minus, so the tussless male genotype form, if it never survives past the earlier stages of embryo development, we would never see it form. Okay, so that is the explanation. And so that's essentially what the scientists figured out was happening. And they also did GWAS essentially to try to figure out what genes are associated with this tussless phenotype. So of course they couldn't do the GWAS on the males that were not being born. They never survived, right? But they were able to do GWAS and look at all the SNPs of this population of elephants to try to find out what SNPs might correspond to the tussless phenotype. And essentially they identified 2 genes. One of them is AMELX. It's a gene, and it's located on the X chromosome, which is what we would expect, right? It's located on the X chromosome, and it encodes for a protein that is involved in biomineralization of an animal. And they also found an autosomal gene. This gene is called MEPA1, and it was found on chromosome 1, and it encodes a protein for dentin mineralization. So you can see both of these genes are biomineralization and mineralization that you might think would be involved in the formation of a tusk, right, of the ivory of the tusks. Okay, so there are 2 phenotypes going on here. One of them is the tussless phenotype, and one of them is this male lethal phenotype. These are 2 different phenotypes. And so we're going to focus first on the tusks. because that one's much easier to try to figure out. So 15% of the female offspring of tuskless mothers have only one tusk. See how this elephant has a single tusk? That's not because it was in a battle or something like that and its tusk got removed or a hunter removed it, like when hunters to kill the animal when they remove the tusk. They're finding like this is a genetic thing. So 15% of the female offspring of the tuskless mothers have only one tusk. And so think about what we learned so far regarding the genetics of the tusklessness. Would all females heterozygote for tusklessness be expected to show unilateral tusklessness? So in other words, have one tusk. Okay, so 73% are saying no, right? 15% unilateral tusklessness be explained by X inactivation alone. Five, 4, 3, 2, 1. Okay, so we got close to a 50-50 or a 60-40 ratio. Okay, so 15% unilateral tusklessness, so that's having one tusk, be explained by X inactivation alone. What is true about X inactivation? What percentage of, if you had a heterozygous tuskless female like this, what percentage of the females would you expect, what percentage of females would you expect if due purely to X inactivation? So what percentage would you think could be tuskless if it's simply due to random X inactivation? Five, 4, 3, 2, 1. Okay, so got a mix between B and C. Okay, so remember X inactivation happens at random, right? You would have each of these chromosomes being inactivated 50% of the time. This one would be the tusk phenotype would be inactivated 50% of the time. The tuskless would be inactivated. Okay, so you would expect to have like a 50-50 ratio, right? It's just random inactivation. You got two chromosomes, so it would be, you would expect it was all other things being equal and being to what you would expect, since it's a 50-50 inactivation, you expect to see 50% of the time that you would have the tuskless phenotype, 50% of the time you would have the tusk phenotype. And that's not what we saw, right? So anyway, that is what we saw, but for the unilateral tuskless, we saw 50%. Okay, so remember embryo development. I talk about this a lot in the course. Actually, I talk about stem cell niches and I talk about the differentiation of cells and how embryo is a little bit different. So embryo has pre-programmed cellular differentiation. Other cells in the body, they get their differentiation signals due to positional information from neighboring cells. But anyway, so in embryo development, you have different cells that are going to give rise to different tissues. And sometimes it's a cluster of cells that together will determine whether an organ or appendage is formed, right? So it could be all of these cells here that become a placenta, or all of these cells here that become the embryo. So there's groups of cells that are going to give rise to particular cell types. Okay, so, and whenever we have exon activation, if exon activation happens, the timing of when it happens is really important. Okay, so the timing is like you have that first cell that's being formed, and it's going to divide and give rise to new cells, and so on and so forth. But eventually, exon activation is going to start. So these orange cells, they turned orange, they begin undergoing random exon activation. And then later, you would have the cells proliferating, and that would give rise to a mosaic, kind of like what you see in the calico cat. Okay, so timing of an exon activation, it typically happens later in the embryonic development, after a certain number of cells are formed. So it's usually, but not always, the first cell that undergoes activation. It's usually after several divisions have already happened that we get random exon activation, and then we get particular cell niches giving rise to a particular phenotype. You can imagine this as like the embryo that's going to give rise to the elephant. And you can imagine that these certain clusters of cells are going to be specified for their role is going to be involved in tusk formation. Okay, so and so since it's random and it's a collection of cells that are going to give rise to the tusked or tuskless phenotype, this can very much affect the timing at which the exon activation happens, can affect the tusked or tusklessness. Okay, so 50% of cells have inactivated the tusk allele. Only cells inside the circle, though, give rise to tusks. Okay, so, and it could be that there's a particular threshold that's going to give rise to the tusk formation or the no tusk formation. So, in other words, if you have like greater than 5 orange, which let's say in this situation, they're going to equal the tuskless alleles being inactivated. So if more than 5 are in that particular niche, you drop below a particular threshold and a tusk would be formed. On the other hand, if you have less than 5 of those cells that have the tuskless inactivated, you would have enough cells there to actually form the other phenotype. So one of them will form the tuskless phenotype, the other one will form the tusk phenotype. Okay, so could 15% unilateral tusklessness be explained by X inactivation alone? What do you think about that? Five, 4, 3, 2, 1. It becomes difficult to I just deleted the responses. Let's do it again. Just to answer this one again. Sorry, I clicked 2 times and it got rid of the responses. Could 50% unilateral tusklessness be explained by X inactivation alone? X inactivation you would expect to be like a 50-50 thing, but anyways, okay, so let's see. After my explanation, still 60% of the class are saying, no, it couldn't be explained by the unilateral tusklessness by X inactivation alone, and 41% are saying it could be. So, what about epistasis, if we take epistasis into account, remember what epistasis is. That's whenever the epistasis is whenever one gene's phenotype or allele composition is going to affect the expression of another gene. And remember what we said here about this whole tusk and tusklessness thing, how ultimately the scientists, they did GWAS and they came down to, they were able to identify two genes. One was autosomal and one was located on the X chromosome. Okay, so that actually does already suggest that there is epistasis. Okay, so anyways, remember we had those two genes. One was on the X chromosome, one was on chromosome 1. And if one gene was affecting, the alleles of one gene are affecting the other gene, could we get this 15%, which is causing the unilateral tusklessness? Okay, so could that be explained by epistasis? So we already know, let's just say that this X-linked gene is the AMELX gene, that, you know, that we were, let's say the AMELX gene is following that X inheritance pattern that we already solved for tusk and tusklessness. And the MEP1A gene, on the other hand, has its own inheritance pattern. It's an autosomal gene, right? So it's going to have its own inheritance pattern. And so how could we get this 15% unilateral tusklessness by considering epistasis? Well, it turns out if you actually if only the individuals that have homozygous recessive alleles for an autosomal gene like MEP1, okay, that would be 25% of the population that should have that particular combination of alleles. And so if you combine that with your tusk and your tusklessness, and ultimately what you would get is 15% of the female individuals, 50% are going to have the tuskless phenotype, times 25% that are going to have the recessive, the homozygous recessive alleles for MEP1. If we take those together, we ultimately would get 12.5% of individuals that share this combination of phenotype, which is very, very close to the 15% unilateral tusklessness. So again, it's not an exact match, but and it still remains unclear. But that's the idea right now, is that since we have those two genes, they both are involved or mapped and thought to be involved in tusklessness. And ultimately, they somehow cause unilateral tusklessness, that it's actually the homozygous recessive individuals for MEP1 combined with those tuskless individuals that inherit one tuskless allele from the AMELX that combine together to give that 15%. Okay, so the AMELX gene sequence is the same among tuskless and tuskless elephants. They actually went in and they thought, okay, we got it. AMELX is definitely our gene. We mapped it there with GWAS, but they found, unfortunately, that the tusked and tuskless elephants actually have the same sequence for AMELX. And so what would be your hypothesis to explain this? No one? Okay. Remember we looked at GWAS and how just because a particular SNP is identified at that particular gene, it doesn't necessarily mean that it's that gene that is responsible. And so it could potentially be explained if there were SNPs in nearby regulatory elements or so on that are outside of that gene that could give rise to the phenotype. And actually, what was found, not in elephant, but a deletion in a syntenic region in humans. That's like the same coding region in humans. A deletion in that region actually causes an X-linked dominant male lethal syndrome. So just like we were seeing in elephants, and that region doesn't just have the AMELX, which is called in elephants, or AMEL, which is found in humans, but also there are other nearby genes. And the deletion of the syntenic region also causes an X-linked dominant, that causes the X-linked dominant male lethal syndrome, also results in in deletion or non-formation of the maxillary lateral incisors. So, you know, these teeth right here, you can see they're missing in this individual right here. So just like elephants, you know, the tusks are essentially formed from those lateral incisors. In human, those incisors are missing as well. So that particular region of the genome definitely seems to have some kind of link to both male lethality and embryonic development, and also to the formation of these particular teeth. Okay, so and so whenever they deleted that particular region in male mice, what they found is that the like similar phenotypes, and the phenotypes could be rescued by overexpressing this gene HCCS. So it wasn't the AMELX gene, it was actually a neighboring gene, and perhaps elements that help the regulation of this particular gene, because whenever you force this expression, you can actually rescue the phenotype. That means you can restore male viability, and you can restore the formation of those particular teeth. Okay, so again, it wasn't due to that particular AMELX gene being missing. It was actually a neighboring gene's expression that was affected. Okay, so the testless and the male lethal phenotypes, those are two different phenotypes. You know, by looking at the genes that essentially encode, you know, there were genes involved in dentin formation and biomineralization. It makes sense that they would be involved in tooth formation of these maxillary incisors and of the actual tusks themselves, but it remains unclear why those same mutations are causing lethality in males. And so I think one thing we need to remember, and this is actually quite common, is that genes often don't have single functions. The same genes that are involved in the biomineralization to form tusks and so on may actually be moonlighting. They may be taking on other roles in embryo formation. Okay, such that whenever they're non-functional, they result in male lethality, even though we only know their role to be involved in biomineralization, tusk formation, and so on. So I think this is a great example to highlight here that genes are often playing multiple roles, and they can seem like, or they can be actually unrelated roles. It's interesting actually in plants, we find enzymes that make particular defense metabolites and stuff, they actually localize to the nucleus sometimes, and they seem to be doing some kind of regulatory function. So they're moonlight. They're actually doing two entirely different roles. Okay, so I just wanted to go back to the example again, because I think this is a concept that people, they often, last year anyways, they seem to struggle with, and that's that timing of X inactivation can affect the phenotype. If X inactivation occurs after cell division, like now, so you have two X chromosomes, they're not being inactivated, and then all of a sudden, later, relatively late in the embryo development, for example, we have the inactivation of the random inactivation of one of the two X chromosomes. So in these 4 cells that I've shown here, then ultimately that would give rise to a mosaic phenotype, right? It would give rise, like in the calico cat, where you would see like, a mosaic. You would see like spotty or blotches of different pigmentation color, for example. In the case of the elephant, that was happening at random, and it's thought that the cells that give rise to the tusks, they need to have a certain threshold limit in order to form of the tusk expression in order for them to form the tusks. If they have the tuskless allele and most of the cells have been inactivated for the tusklessness, they'll form the tusk phenotype. If they have more cells that are inactivated and for the tusk phenotype, then they'll have the tuskless phenotype. And the tuskless phenotype is dominant in the early stages of development. Okay, so now that's if X inactivation happens later. If X inactivation happens very early in development, what do you think you would find? So what if the very first cell actually undergoes X inactivation and all subsequent cells are maintained? What do you think you would see? Would you see a mosaic phenotype? Or would you see, what would you see? Okay, so, yeah, go ahead. Sorry, anyone who raised their hand, go ahead. Christian?
Possibly, sorry.
Daenerys? Yeah, no, go ahead, Daenerys, go ahead.
I think that probably this cell will lose to like multiply because if there is no X chromosome, for example, if it is like a male cell, right, it will actually have only Y chromosome and it will be inactivated. If it is a female, it still have only one X chromosome and it will undergo these several mutations that may be unviable.
Oh, okay. Yeah, but remember, so whenever there is one X chromosome and a Y chromosome, like in the males, the X chromosome doesn't get inactivated, right? So here we're talking about a female, and it has 2 X chromosomes, and one of them is going to be inactivated. What would you expect to see? Christian?
Yeah, so because this is one of the earlier cells of development, because we would have inactivation of one of the X chromosomes through mitosis, these epigenetic changes, right? The activation of, the exist loci for whatever X chromosome is inactivated, that would be propagated through the daughter cells and the lineage from the first cells. So we'd likely see less a variation with respect to, tuskless or tusked. We'd either see more tuskless or tusked cells.
Right. So, very good. So, if inactivation happens later in development, let me go back here. So, if the cell it's expressing, this blue cell here is expressing 2 X chromosomes. And it gives rise to another cell that's expressing the 2 X chromosomes. And these cells are all expressing the 2 X chromosomes. And then after forming numerous cells, random, I'm drawing the 4, they look pretty symmetric, but this would be happening at random, right? So now all of a sudden, after forming a certain number of cells, you would have random X inactivation, and that would ultimately give rise to this mosaic phenotype. Now, you have the X inactivation happening right at the beginning. That first cell, upon being formed, actually undergoes X inactivation. It's not frequent that that happens, but it is possible, and some species is more possible than others. You essentially would give rise to all of the cells having exactly the same phenotype. Because, like you said, it would be they would be inherited in all of the subsequent cells. The same X chromosome would be inactivated in all of the subsequent cells. Okay, so that's what would happen if it was in early development. Does that make sense? I hope so. I can't remember what else I wanted to say about that. If this, here I'm drawing this, the blue cell, one chromosome gets inactivated and it goes to the orange cell, and then all subsequent cells are orange. But what would you expect if X inactivation happened in the very first cell, and you had a heterozygous genotype, so like the X plus and the X minus? What would you expect to see at the population level? So assuming that this X inactivation happens at random and it happens in the very first cell. Let's say there's 2 possible phenotypes. There's the phenotype of the X plus and the phenotype of the X negative. So what would you see at the population level? Somebody want to raise their... Kristen? Go ahead.
Yeah, so if we're considering this case here, for any individual, they're likely to have one phenotype or another. But comparing individuals within a population, the tuskless versus tusked phenotype in the cells would likely be variable.
Okay, so that's an added complexity, right? But yeah, go ahead. Ahuna?
Because it happens in the early cell, there's a 50% chance of X inactivation in every cell. So for every new offspring, they're just as likely to have either X plus or X minus inactivated. So if an individual X plus is inactivated, then that individual would only have X minus cells. So that individual would be tuskless. And it's just as likely for the reverse to happen, where the individual would have all X plus cells. So at the population level, you would expect to see 50% tuskless individuals and 50% tusk individuals.
Right, and yeah, exactly. So I'm trying to stray away from the tusk phenotype. It can be a little bit confusing, but I guess whatever this phenotype encodes, the calico cat, it's either orange fur or black fur, I guess, right? And so you could have, if the X inactivation actually happens in the very first cell, rise to all other cells. Essentially, if it were the orange fur color X that got inactivated, all of the cells would then be black color, right? All of them would inherit the black chromosome as functional, and the orange fur color chromosome as being inactivated. So that entire cat would be black, have black fur color. On the other hand, if the cat inactivated the very first cell inactivated the black chromosome, then you would get your orange phenotype, and all cells that were derived from that cell ultimately are going to be orange color. So imagine that happens early, very early, and it happens in the very first cell stages of embryogenesis. You can have an individual that has an entire solid color. It might be black, it might be orange. Whereas if it happens later in development, we actually get something like this. We would get the mosaic color. Okay. That's all I have for today. Again, your deep questions come out tonight, 7 p.m. If you have any questions about this or other materials, let me know. Other than that, I will just finish class a little early today and get some rest. If you have questions, though, by all means, just let me know. If not, please remember that Thursday is a session for just working on your final deep questions, guys. Okay, so I'll see. I don't know if I'm going to be able to come in person, but if not, I'll try to do a Zoom thing again so you guys can ask questions. Dinera, you have a question?
What about the worksheet of today's session? Do we need to do that on our own and then turn it in?
You know what? The worksheet, yeah, because you can see it on eClass, right? I actually removed it the last minute because I was actually afraid that we weren't going to have enough time to get through everything. So you don't even need to do it.
Thank you.
Yep. Okay, guys. Okay, I will talk to you on Thursday, either over Zoom, or if I'm feeling spectacular tomorrow and the next day and I'm 100% convinced that this is cleared, I may come to class. Okay? All right. See you. Oh, Anoyola, you have a question?
Yes, sir. I would have sent you another email. I'm not sure if you saw it. I saw that you replied to me yesterday.
Huh?
I'm not sure if you saw my last email that I sent.